Our neural response to speech starts with two types of nerves in the eighth cranial nerve. One set of high threshold neurons only respond to a higher level of stimulation, so a higher intensity sound. These neurons generally don't fire unless there's a high intensity sound present. Lower threshold fibers respond to much quieter sounds at low signal intensities, and they also display a certain amount of random firing even when there is no sound signal. This helps us process sounds that are near the minimum threshold of intensity of sounds we can process. These two different types of fibers are similar to some of our other senses. The high threshold neurons are more like our sense of touch. It doesn't generally respond unless there is something actually happening. The low threshold fibers are a little more like our sense of temperature, where they have a continuous firing rate that gets altered by stimulation. We understand sound intensity by a combination of what types of fibers are firing, just low or a combination of high and low, as well as their firing rate, where faster firing is encoded, used to encode louder sounds. Neurons generally have a characteristic frequency, a particular frequency of sound that it responds to best, and this is represented by what's called a tuning curve, as you can see in this image. It gives you an idea of how much sound pressure is required for the neuron to fire at different frequencies of sound. The frequency specificity of the basilar membrane affects the tuning curve for the neurons. The lower frequencies basically have a finer response compared to the higher frequencies, and we can quantify this reasonably well in terms of looking at percentages of change. So if you're up around 10,000 hertz, a 1% change is a 100 hertz change, whereas if you're around 100 hertz, a 1 hertz change is a 1% change. We then go from the individual responding neurons in the auditory nerve part of the eighth cranial nerve to further areas of processing in the auditory system. We have both temporal and tonotopically, which means by frequency arrayed information that gets passed to progressively higher centers in the neurological system for extraction of information. Within the cochlear nucleus, which is our first processing point outside of the cranial nerve, we have a variety of different types of nerves that respond to sound stimulation in different ways. So-called primary-like neurons respond similar to our cranial nerve neurons, have a very frequency-specific response to sound coming in. Onset response type neurons fire when there is initial stimulation of sound, and they have a broader tuning to a range of frequencies, but after that initial stimulation, their response drops off. Chopper response neurons respond to sounds that are ongoing. They are also less frequency specific. Pauser response neurons are sort of like onset neurons, but only for the highest intensity sounds, which will cause them to fire, and then they won't do much else. Build-up response neurons gradually increase their firing rate as the sound is ongoing. There are also more complex cochlear nucleus responses that happen. There are some that produce inhibitory responses, basically firing when the stimulation isn't right for them, so to speak. These come mostly from the dorsal cochlear nucleus, and they help narrow the specificity of the frequency response of the system. There are neurons that are sensitive to changes in frequency, either from low to high or high to low, so they can detect what's called frequency modulation in the sound. There are also neurons that are sensitive to changes in intensity, things getting louder or things getting quieter, which is known as the amplitude modulation of the sound. As we leave the cochlear nucleus and get to the superior olivary complex, this is where we localize sound in space, so information is processed that helps us tell where a sound is coming from. In the lateral superior olive, we compare the intensity of sound between the two ears for higher frequencies of sound to localize. So the sound is going to be a little louder on the ear that is closer to the place the sound is coming from. In the medial superior olive, we compare time patterns of the changing sounds known as phase differences, mostly for low frequency sounds. So the idea is that the sort of waves of positive and negative pressures of the sound are going to hit the two ears at slightly different times. And so we can use that difference in phase times to determine whether the sound is coming from one side of the head or the other. The inferior colliculus receives information from a lot of different locations, the cochlear nucleus, the lateral lemniscus, and from the superior olive. And this appears to be the location where we make the finest frequency sensitivity responses to the sound, so really narrow the response down to specific frequencies that are in the sound. The medial geniculate body in the thalamus then gathers up all of these neurological responses and passes them along to the cortex, primarily to Heschel's gyrus, but also to other areas in the temporal lobe, as well as to association areas that are in the parietal lobe that connect sound up to our other senses. Arrival of sound information to the cerebral cortex comes primarily from the contralateral ear, the ear on the other side, through the thalamus. The bulk of the information first goes into Heschel's gyrus, which is our core of auditory processing and the primary receptive area for sound. Surrounding that core area is the belt, which helps clarify the distinctive aspects of the sound input and tune out random aspects, random parts of noise that are part of the response. The parabelt provides further detailed processing of that information that then gets sent out to the other parts of the brain. Responses within the brain happen through a variety of interconnections and in a variety of areas. Some of these areas are bilateral, which are indicated here. So we have acoustic analysis in terms of frequency and time in the green spectrotemporal analysis box, marked with a similar green in the auditory reception area on the temporal lobe on the brain. We have phonological analysis associating these spectrotemporal frequency and time components with individual speech sounds. In the yellow area, so in the kind of box diagram indicated as the phonological network, and on the brain image in a more central part of the temporal lobe. We also have that information then getting associated with words and meanings in a lexical and conceptual analysis, indicated in the pinkish purple, a little more laterally along the temporal lobe. In addition to these bilateral responses, we then have a certain number of what are typically left side dominant responses in people, though in some folks the percentage is small. This can be switched to the right side. The odds of that happening are higher if you're left-handed. We have more contextual processing that happens in the anterior part of the temporal lobe that helps take these take this acoustic information and this sound information and consider it in the particular context in which it was produced. We have automatic association of the sounds we hear with articulation, how we would say the sounds, and with the literacy system, and on the production side, that would be the writing side of things in Broca's area and in the motor strip. We also have association of these sounds with higher level structures in syntax in morphology, and also with our literacy areas in terms of how we would read these things in Wernicke's area. These higher level, more language processing areas are indicated in blue in the frontal lobe, and in the association area, kind of where the parietal lobe and the temporal lobe and the occipital lobe are all intersecting at their boundary. So an overall summary of the physiology of hearing: we have our mechanical responses in the outer and the middle ear that funnel acoustic information and translate it into fluid motion. The inner ear, we take any acoustic information and convert it into spectral and temporal information about the sound, which then gets passed along through various higher levels of neurological processing to associate these things in speech with things like our lexicon, with our grammatical structures, and with our literacy skills. There's also shared anatomy in the inner ear and the neurological system with the vestibular system that will tell us things about our head posture and our body movement.